So hi, everyone. I'm Gil. I'm a PhD candidate in the University of Vienna. And today I'm going to talk about a comparison that we did between two bait sets to studying a rapid radiation. This work has been recently published, so I have the link here, but I will give a short overview of what we've done. In my project, I'm using genomics to study a non-model group, and we became very interested in using target capture. It's a very popular method, and in target capture, small probes are used to hybridize to known DNA regions, and everything else in the DNA library is washed out. So after sequencing, what we have are the same genomic regions from all our individuals. When we were planning this project, we found ourselves with a question that we think other groups also face, whether we were going to create our own taxon-specific bait set. In our case, it's a bait set for bromeliads. And creating your own bait set is time intensive and requires resources like a reference genome. And on the other hand, we could use a universal bait set. In this case, the Angiosperms 353 set. It's a bait set that targets 353 regions that are highly conserved among all flowering plants, and it's been successfully used to resolve phylogenies in many different plant groups. Of course, we assumed that if we use a universal bait set that is more cost effective, it's going to cost us less time and money. When we target regions that are highly conserved, maybe we will not have the same resolution power, maybe we will have fewer markers and we will see differences. And we decided to compare them. And this was the basis of the work. I'd like to present our research system, the Bromeliaceae family, a family of monocot neotropical species. Probably the most famous member is the pineapple, the Ananas squamosus. The members of the family are characterized by a typical water tank. They have a structure where they impound rainwater within a rosette of leaves. It's a very diverse group. We find them in different ecological niches, from humid rainforests to arid environments. And in my project, I'm specifically interested in the genus Tillandsia, subgenus Tillandsia. It's a very diverse group that radiated quickly. What we know about its history is that the Bromeliaceae family probably originated in the Guiana Shield around 100 million years ago. The subfamily diversified in a way that is correlated with the uplift of the Andes. And the subgenus Tillandsia expanded into Central America around 3 to 2 million years ago with a closing of the Panama Isthmus. And members of this group are mostly epiphytic, and they are very diverse. We see different adaptations to their habitats. We see repeated shifts of photosynthetic syndrome, both CAM photosynthesis and C3 photosynthesis, different pollination syndromes, and these are considered key innovations, which means that they accompany diversification in the genus. We're studying a rapid radiation, which means that we're studying speciation through time, and we're interested both in shallow timescales and deep timescales. So I'm going to talk about phylogenomics today and a little bit about population structure and population genomics. So we started by designing our own target capture kit for bromeliads. In this kit, we included single copy protein coding genes. We also included genes that, according to the literature, are associated with the key innovative traits that we want to study. We included genes that were previously used as phylogenetic markers and also orthologs to the universal kit. Our kit targets 1,776 genes, so we named it the Bromeliad 1776. For the comparison between the two kits, we sampled 72 samples from the whole Bromeliaceae family. We included 40 accessions from our focus group, the rapid radiation of Tillandsia. We performed 2 enrichment reactions, one with each bait set, and after sequencing, we reference mapped them back to the Ananas squamosus, and we call variants. When we first looked at the data, what we found was that the mapping rates were much higher for our taxon-specific kit. We saw a higher percentage of reads on target for the taxon-specific kit, and that means that we got more sequences that match the probes that we designed. With the universal kit, we had markers from random regions across the genome. The depth, the coverage of the data was almost twice higher for the taxon-specific kit, and we also had 4 times the number of SNPs. So far, this is what we expected. And then we looked at phylogenomics, and it's a big slide, so I will walk us through the different parts. This is a coalescent-based species tree. For this tree, each gene was considered a genomic window. We see on the left the bromeliad taxon-specific kit, and on the right, we see the universal kit. All the branches have the posterior probability support of 1, unless noted otherwise. And the main findings that we saw in our phylogeny was that the topologies we retrieved between the 2 bait sets were not very different. We saw differences in the placements of a few species, but they were not big. And we didn't see much difference for the clades where we had very thorough sampling. The branches were pretty well supported in both bait sets. We see some internal branches with lower support with the universal bait set. I also want to say that the results were quite similar for maximum likelihood inference. Where we did find differences that are interesting to talk about was that when we looked at gene tree incongruence. And here we see on the pies the proportion of trees that agree with the main topologies, that's in blue. And we see the proportion of genomic windows that agree with the top alternative and all other conflicting topologies in green and in red. And in gray, we see the proportion of trees that were non-informative for this analysis. And we can see that with the universal kit, the proportion of these trees was much higher. We had many windows that were just not informative. So we wanted to take a closer look in these windows and see why this happened. And here we see on the left the length of the windows. We see in blue the length of the size, the length of the windows that we targeted, and in red we see the length of the windows that we captured. And on the right we see the average gene tree bootstrap support. And what we see is that the length of the windows that we targeted and also that we captured is much, much higher for our taxon-specific kit, more than twice the size. And the average gene tree support was much, much lower for the universal kit. The average was around 10, which is very low. And this is, and this is a finding that we expected from targeting regions that are highly conserved, that they are not going to be as informative. As I said, we also wanted to perform population genetic analysis. So here I have the inference of population structure that we did using admixture. And again, we see the 2 different sets. And in general, we didn't find a strong population structure. We were able to retrieve the population of the different species, but that's about it for our tillandsias. And we see different values of K from K2 all the way down to K7. So again, we didn't find big differences. You can see it in the colors. The 2 kits performed comparatively well, even though our taxon-specific kit had 4 times the number of markers. Where we did find some more interesting differences is when we tried to find the best value of K that it was most highly supported. And this we did using a cross-validation error method. And on the left is the universal kit. On the right is our taxon-specific kit, and we can see the lowest cross-validation for K5, and in our taxon-specific kit, it was quite easy to find the best value of 5, and with the universal kit, it was not that obvious, and it was very tricky. So I was a little bit fast, but we're going to conclude. What we found is that both kits, the universal and the taxon-specific, performed comparatively well. We found a small advantage for the taxon-specific kit, especially when we looked at gene tree incongruence. And we think it makes a bigger differences if you're studying lineages that are rapidly radiating. I also want to say that we found differences in the data filtering. The universal set, the data that we had with the universal set, it was more challenging to achieve a data set where we had high confidence in. For groups that are considering using target capture for their own research, we have some take-home message. Generally, if what you want to do is describe an evolutionary history, then universal kits are excellent. They offer excellent opportunities to do phylogeny and beyond that. If you're interested specifically in gene tree incongruence, and especially if you're working on young, rapidly radiating groups, then a taxon-specific does have advantages. That being said, we designed our kit to do more than just phylogeny. We designed our kit to study molecular evolution and trait evolution in tillandsias. And there are a few ongoing projects in this group studying photosynthetic shifts. So in that case, if you design a taxon-specific kit, you can design one kit and use it for multiple purposes and perform different kinds of research with just one data set. So with that, I'm going to finish, and I'm going to thank all our co-authors for the fantastic work that they put into this project, my supervisors, the organizers of this event, thank you very much. And thank you everyone for your time. I would love to take questions if there are any.